Hello student, all of you are most welcome on my channel, that is Liberty Solution. In this video, I will be discussing about chapter number 11, Biotechnology, Principle and Processes Part 2. Like, comment, share it with your friends, and don't forget to subscribe my channel to get many more videos. These videos are useful for your school examination, board examination, as well as NEET examination. So let us start. Biotechnology, Principle and Processes, Part 2. So in Part 2, I will be discussing about process of recombinant DNA technology. Now, what is recombinant DNA technology? So there are many steps in this recombinant DNA technology. And that is, first step is what? First step is isolation of genetic material, that is DNA. Second is cutting of DNA at specific locations. Third, cutting means DNA is very large molecule. It is very difficult to handle. Therefore, we have to cut into small pieces. At what specific location? So this is second step. Third, if you have got only one small piece, suppose this one. So in case we have misplaced, then again we have to isolate. So we have to get what? Get multiple copy of this desired fragments of DNA. That is called amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. PCR stands for polymerase chain reaction. To get what? Multiple copy of required desired DNA fragment. Then this DNA fragment need to be what? Inserted. So inserted in what? Host cell. So fourth step of recombinant DNA technology is insertion of recombinant DNA into the host cell. Then fifth step is obtaining the foreign gene product. What is the product of this gene of interest? We have to obtain that product. Then we have to do what? Downstreaming process. That include what? Purification, filtration. That are the part of downstreaming process. So we'll be discussing one by one. So these are 6 steps of recombinant DNA technology. Sometime in the question paper, there will be there in CBSE board examination, every school examination, there will be question explain what are the various steps of recombinant DNA technology. So you have to write these 6 steps. Now first, isolation of genetic material, that is DNA molecule. Suppose this is a bacteria. So you can isolate DNA from bacteria. You have to isolate DNA from plant. You have to isolate the DNA from what? Liver of animal. So from here, we have to isolate genetic material. So whatever your sample may be, treat bacterial cell, plant or animal tissue with enzyme like lysozyme. If it is a bacteria, you have to treat this sample with lysozyme. If it is a plant, then you have to use what? Treat it with cellulase. And if it is animal, you have to, like fungus, you have to treat with chitinase. So why? To break down their covering, external covering. So lysozyme is used for bacteria, cellulose is used for plant, and chitinase is used for fungus. Sometimes question will be there in column A and column B. You have to match A with B. So now the cell is broken, releasing DNA and other macromolecule like RNA, protein, polysaccharide, and lipids. So if you are smashing these material by treating lysozyme, cellulose, and chitinase, then cell will be broken, and whatever is inside, there may be DNA or other macromolecule like RNA, protein, polysaccharide, and lipids, they will be coming out. Then RNA is removed by treating ribonuclease. So there may be RNA, so you can remove RNA by treating what? Ribonuclease. Proteins are removed by treating protease. It will get digested. Other molecules are removed by appropriate treatment. When chilled ethanol is added, purified DNA precipitate out as a fine thread in the suspenser. So the material is here. If you are adding chilled ethanol, it will get precipitated. So this is what? Isolated DNA. Separated DNA can be removed by spooling. This is what? Spooling. You may have done this experiment in your school laboratory. Now, purified DNA is incubated with restriction enzyme. So what is the function of restriction enzyme? As a result, DNA digests. If you are cutting it, you are treating with restriction enzyme, large DNA molecule will be broken down into small fragments. So this is a large molecule, so it will be broken down in small fragments. So this is DNA sample. Then you are treating this DNA with what? Restriction enzyme. It will be cut into small pieces. Now restriction enzyme cleaves DNA into smaller segment of various size. So now DNA fragments are separated by a technique called gel electrophoresis. This is a technique, gel electrophoresis. So what is gel electrophoresis technique is used? This is a technique used to what? Separate DNA fragments as per their length. So here there is a gel electrophoresis. There is one technique called agarose gel electrophoresis. Agarose gel electrophoresis is employed to check the progression of restriction enzyme digestion. So this is agarose gel. Here, this is what electrode is there. Electrophoresis, there are two electrodes are there, positive anode and cathodes. So there are some wells are there, 1, 2, 3, 4 wells are there. These are the wells. These fragments are loaded on these wells. Then under the influence of electric field, they start migrating because DNA is what? Charged particle will start migrating. And here this gel will be having what? Small size, pores will be there. So those which are having larger in size, they will be traveling less distance, and those which are smaller, they will be traveling more distance. So gel electrophoresis showing a migration of undigested lane 1, undigested set of DNA fragment, that is lane 2 to 4. Here there is no migration in lane 1. Lane 2, 3, 4, there is what? Fragments are there. So they are what? Digested. Second step is cutting of DNA at a specific location. DNA is negatively charged. DNA is what? Negatively charged. So it moves toward the anode. DNA fragments are separated according to their size through sieving effect of agarose gel. This agarose gel is obtained from polymer from seaweeds, Gracilaria, Geladium, you may have been knowing. Smaller size fragments move further. So if this fragment is small, it will be traveling more distance. And if fragment is large, they will be traveling less distance. The process is repeated with vector DNA also. DNA fragment can be seen as a bright orange colored band when they are stained with ethidium bromide and exposed to UV radiation. In simple light, we cannot see them, but if you are exposing these agarose under UV radiation, so they will be color will be what? Like bright orange. And we have before watching them, seeing them under a UV microscope, we have to treat them with ethidium bromide. So sometimes the question will be there, they name the chemical which is used to stain DNA fragments. So answer will be what? Ethidium bromide. So what is the color of this DNA fragment? Bright orange. They are seen under which microscope? Ultraviolet radiation microscope. Light microscope cannot be, we will not be able to see these bands under light microscope. DNA bands are cut out from the agarose gel. This is called elution. Here, this is what? Here, bands are there. They can be cut, and this is called what? Elution. Excised DNA band and A2 gel dissolver buffer. The cut out gene of interest from source DNA and cut vector are mixed and ligase is added. It creates what? Recombinant DNA. So in this way, this is what, suppose this is the desired fragments of DNA. Then we will be separating it. There may be many materials, so we'll be separating. Ultimately, we'll be getting what? DNA fragments. So this gene of interest is here. So in this way, this gene of interest will be joined with the vector DNA by the enzyme called DNA ligase. So ultimately, we'll be getting what? Recombinant DNA. Recombinant DNA means gene of interest DNA plus vector DNA, that is called recombinant DNA. Now, third step is amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. There is a machine, PCR machine, that is having thermal cycler. It is also known as thermal cycler. Polymerase chain reaction is the synthesis of multiple copies of gene of interest in vitro. In vitro means artificial medium, using two set of primers and the enzyme DNA polymerase. Here we need two primers and enzyme DNA polymerase. Primers are small. What is primer now? Question, what is primer? Primers are small chemically synthesized oligonucleotide. They are oligo means few. Nucleotide means you know already nucleotide. Like polynucleotides, here they are oligonucleotide that are complementary to region of DNA. Now we are doing what? Amplification of gene of interest. What are the steps of PCR? Denaturation, annealing, and extension. These are what? Sometimes question will be there. What are the steps of PCR? So denaturation, annealing, and extension. What happened in denaturation? See, double-stranded DNA is there. This is what double-stranded DNA is there. This region need to get what? Multiple copy, this region. So first we have to do a denaturation. In the denaturation, we have to heat this DNA. So these band, these two strands, they will get separated. So then that is, next is what? Annealing. Next is what? First annealing. So here annealing, what happened? Primers are added. Two set of primers. This primer and this primer plus enzyme DNA polymerase. Primers, what are primers? Primers are nothing, they are what? Oligonucleotides. They are oligonucleotides. Then after annealing, they are, third step is extension. What happened here? There is a DNA polymerase enzyme is added. It is isolated. It is also called tuck polymerase because it is obtained by Thermophilus aquaticus. Tuck stands for Thermophilus aquaticus polymerase plus deoxynucleotides are added. Then there will be extension. These remaining part of the strand will be synthesized. It is synthesized here. This is called extension. Then it is this process is repeated 30 times. Though then we can get what? Millions copies of this desired DNA. So this is called what? PCR, polymerase chain reaction. So in the PCR, there is first step is what? Denaturation. What happened here? It is the heating of target DNA, gene of interest at high temperature. How much degree? 94 degrees Celsius to separate the strands. Each strands act as template for DNA synthesis. Now these separated, they will be working as a template for DNA synthesis. Then second step, annealing. It is the joining of two primer at 52 degrees Celsius at 3' end of DNA template. So primers are added at 3' end. Here, this 3' end, they are added. This is 3' end, this is 3'. So they are added at 3' ends. At what degree, at what temperature? 52 degrees Celsius. Then third step is what? Extension. Third step is what? Extension. What is extension? It is the addition of nucleotide. We are adding what? Nucleotide, deoxynucleotide to the primer using a thermostable DNA polymerase called Tuck polymerase. Tuck polymerase is isolated from a bacterium Thermus aquaticus. Sometimes question is there, what is Tuck polymerase? This is enzyme which is used in what? Extension step of the PCR isolated from Thermus aquaticus. It is a bacterium. It remains active in this Tuck polymerase is active at high temperature during the denaturation of double-stranded DNA. Through continuous replication, the DNA segment is amplified up to how many times? 1 billion copies are obtained. Now, ultimately, the amplified fragments can be used to ligate with a vector for further cloning. So, PCR at a glance. This is a DNA sample. Then there is a primers are there. Nucleotides are required. Then PCR tube is there. Buffer mixture is there. Tuck polymerase are there. They are added here. Then they undergo these processes: denaturation, annealing, and extension. Denaturation takes place at 95 degrees Celsius. So here strand separates. Then at 55 degrees, primers bind templates. These are primers. Then at 72 degrees Celsius, synthesis of new strand. That is called extension. This is all about what? PCR. Then next step of the PCR is insertion of recombinant DNA into host cell or organism. Now we have got what? Recombinant DNA. Using any method, the ligated DNA is introduced into recipient or host cell. They take up DNA from its surrounding. This recombinant DNA is there. Now this is a host cell, E. coli. It is not having recombinant DNA. So here this host cell will be exposed to this what? Recombinant DNA. Now this cell will pick up this recombinant DNA. So now this will be called what? Transform cell. So here there will be some transform cell, some will be non-transform cell. Then they will be transferred in nutrient medium containing ampicillin. So those which are having ampicillin resistant colony, they will survive. Those which are not, now it will be having what? Ampicillin resistant. So it will not be having ampicillin resistant. This one non-transformant will not be having ampicillin resistant. So if you are keeping them, they will die here. And only ampicillin, the E. coli which are having ampicillin resistant, they will develop their colony. So if a recombinant DNA bearing ampicillin resistant gene is transferred into E. coli cell, the host cell will become ampicillin resistant cell. If the transform cell are spread on agar plate containing ampicillin, only transformant will grow and transformant recipient cell will die. So in this way, we can identify what is transformant and what is non-transformant. Now the ultimate aim of recombinant DNA technology is to produce a desirable protein. So this is what target gene construction of expression plasmid. Then these are mammalian cells. This cell, they will be producing recombinant protein. If a protein encoded foreign gene is expressed in heterologous host, it is called recombinant protein. So now the bacteria, a mammalian cell, they will be producing the protein of foreign DNA. So that protein will be called what? Recombinant protein. Cell with foreign gene can be grown in laboratory. The culture are used to extract desired protein and purify it by using separation technique. The cell can also be multiplied in continuous culture system. Here the used medium is drained out from one side while fresh medium is added from the other. It maintains the cells more physiologically active and so produce a large biomass. It yields more desired protein. So there is a continuous culture system is required for the growth of this recombinant DNA product. Now, there are some bioreactors. These are bioreactors where we will all these processes will be taking place. These are the vessels in which raw materials are biologically converted into specific product, enzyme, and other materials using microbial, plant, animal, or human cell. Bioreactors are used to produce large quantity of product. They can process 100 to 1,000 liters of culture. A bioreactor provides optimal growth condition, optimum temperature, optimum pH, substrate, salt, vitamin, oxygen to get our desired product. Most commonly used bioreactors are stirred tank reactor. So they are what? Stirred tank bioreactor is stirred tank bioreactors. These stirred tank bioreactors are two types, simple and spurge. Simple stirred tank bioreactor and spurge stirred tank bioreactor. It is usually cylindrical or with a curved base to facilitate the mixing of reactor content. The stirrer facilitate even mixing and oxygen availability. There are stirrers out there. These are stirred. They help in what? Proper mixing. Alternatively, air bubble can be bubbled through the reactor. In spurge stirred tank, air bubbles are passed to for what? Proper mixing. So, if you see, there are some parts of our reactor, and agitator system is there. Here, one agitator system is there. This flat bed impeller. Oxygen delivery system is there. Foam control system is there. Temperature control system there. pH control system is there. Sampling port is there. So these are different parts of bioreactors. Now, then next is what? Downstream processing. Now, what do you mean by downstream processing? This is what? Upstream processing. Here there is a downstream processing. It is a series of processes such as separation and purification of product after biosynthetic stage. The product is synthesized, but there may be so many impurities. So what is downstream processing? It is a separation and purification of product. The product is formulated with suitable preservative. Such formulation undergo clinical trials and strict quality control testing is done. Here, cell number O2, time, RPM, pH, nutrient medium, flux rate, all these things are under control. That is called upstream processing. And there is our downstream processing. So what is analyzed? safety, efficacy, recovery, cost, quality control, all these things are managed during downstream processing. So in this way, this chapter is completed. Thank you very much for watching my video.